My name is Andrew Scott and I'll be presenting on the PET journey, trials, evidence and approval. As mentioned by the previous speaker, Dale Bailey, clinical PET started in Australia in 1992, 30 years ago. Back in 1992, the initial clinical indications for FDG PET were for myocardial viability, epilepsy, and in some glioma indications. At that time, and certainly with the applications to the Commonwealth and state governments for funding, these were the clinical areas which were emphasised, which would most be impacted by PET being available. However, at the time that PET did start in Australia, there was emerging evidence in the literature that there was an increasing role for FDG PET also in other cancer types. And certainly in 1991, a very famous front cover of the Journal of Nuclear Medicine, you can see in this slide, suggested that clinical PET, its time has come. At the two sites that were initially doing PET scans in Australia, at the Austin Hospital and Royal Prince Alfred Hospital in Sydney, increasing numbers of oncology patients were being evaluated in very specific areas to try and see whether or not the overseas data that was being generated in single site studies was in fact being observed here in Australia as well. And that was certainly reflected in the numbers of scans which was performed at the Austin Hospital between 1992 and 1994, which is shown in the slide, where in the first year of operations, you can see that the predominant number of scans performed was in neurology indications, cardiology 25%, and only 14% of patients were cancer patients. But by 1994, you can see that oncology patients and neurology patients both can constitute 41% of the total number, with only 18% being cardiac studies. And this percentage of oncology patients dramatically increased over the subsequent years. Both at the Austin, at the Royal Prince Alfred Hospital, as mentioned, the numbers of PET scans was increasing with time. And around 1996, two new PET sites were being announced and established in Australia, one being at the Peter MacCallum Cancer Centre by Rod Hicks, and the other at the Wesley Hospital in Queensland by Ken Miles. In addition, for those who were not able to get PET scanners, which were very expensive at that particular time, coincidence gamma cameras were being developed and sold. And a number of sites were starting to perform coincidence PET with gamma cameras with thick crystals, so that they could actually also look at FDG studies in some patients as well. In the early 90s, the scanners, which were identical at both the Austin and Royal Prince Alfred Hospital, were an ECAT 951R system. And this is the type of imaging that we would obtain on the left-hand side. Of course, remembering they were PET-only scanners, and these are simply the CTs that were performed on a standard CT scanner. And you can see in this coronal image going from the neck down to the mid-abdomen, there's a lesion here on the right side of the hilum, which corresponds to a primary lung cancer. And there was also evidence of FDG uptake just in a precoronal lymph node, which you can see just here. This scan actually took 60 minutes to acquire with the patient on the scanner, which meant you could only do 5 to 6 patients normally per day as a maximum. Of course, technology's moved on a lot since this time, but the scans did take a considerable amount of time, also because of the limited field of view as well as the sensitivity of the instruments. It wasn't just FDG PET that was being performed in the 90s. If a cyclotron had both proton and deuteron abilities, you could actually generate additional isotopes like N13, C11, and particularly O15 with a 2-minute half-life. And at the Austin, we were actually able to generate O15 and then be able to send this gas through to a converter, which you can see here just underneath the scanner gantry, which actually could convert the O15 gas into O15 water, which could then be injected into the patient in response to certain activation stimuli. Here you can see a monitor, which is just above a patient who's in the gantry of the ECAT 951R system. And when a particular stimulus would come up on the monitor, you'd actually flick the switch, the cyclotron would send the gas through, and you'd actually infuse the patient, and you could actually identify which parts of the brain were actually responding to the stimulus that was present on the monitor. These obviously predate the current, more commonly performed MR-based activation studies, but they're really quite transformative at the time, and quite a number of research studies and grants were obtained using this technology. Funding for PET became increasingly important because a lot of the initial funding was really focused on CapEx and the establishment of the PET sites. And to be able to run a PET unit and to pay for FDG or to run the cyclotrons, employ the staff, you need to have ongoing funding. So there were multiple submissions for recurrent funding to the Commonwealth Department of Health during the early 90s to try and establish a more regular way that they could actually contribute to the ongoing operational funding. This was particularly problematic at the Austin, where our operational budget was halved within a 2-year period. And so we were delighted in 1997 when we were finally able to achieve a 3C determination for funding for FDG PET, which was written at that time by, or signed off by Michael Woolridge, the Minister for Health, which allowed for funding for FDG PET scans that were performed at the Austin Hospital and the Royal Prince Alfred Hospital specifically. And that was a 4 to 5 year journey to actually get that funding. But there were further submissions to the Commonwealth Government for expansion of the funding, because as I mentioned, there were 2 other sites which had just commenced operations with FDG, which was actually at the time provided from the Austin Hospital principally. Also, in 1998, there was initial funding approval for FDG PET in a reasonable number of cancer indications by the US Medicare agency. And so that further increased the evidence for governmental acceptance of FDG PET. And so in 1999, there was a further consideration by the Medicare Services Advisory Committee, or MSAC, of PET. And this led to a formal review in 2000 by MSAC, as well as state government reviews of PET, to determine whether funding should be provided in an ongoing way. The Commonwealth Government MSAC review included input from PET experts from around the country. And so there was an expectation at the conclusion of those initial meetings that there would be a reasonably positive response from the Commonwealth Government for funding. However, we then learned that the key findings of the final report that was issued by the Commonwealth Government was that general implementation for PET was not justified at that time, and that a limited implementation of PET was proposed with associated limited Medicare funding. This was actually subsequently initiated with a declaration of Medicare funding at the end of 2001, but which came into effect in 2002. And it was proposed that 7 sites be funded around the country, 1 in Western Australia, 1 in South Australia, 2 in Victoria, 2 in New South Wales, and 1 in Queensland. So very much based on population. But that these sites could only receive Medicare funding if they undertook a data collection process, which was to be called the Commonwealth PET Data Collection Project. But this was very surprising for those of us who'd been on that initial committee. Firstly, because this wasn't really consistent with what we understood the outcomes from that particular review were going to be. The second point was that it wasn't clear exactly what data needed to be collected, because the commentary at the time was that they wanted to look for improvement in patient outcomes, which is clearly not achievable for a simple diagnostic test. And so it was unclear exactly what sort of data was required in order to finally get formal Medicare approval for relevant indications, and nor how this was actually going to be run and operated. In 2001, tenders went out for those 7 sites around the country. They were awarded to the Wesley Hospital in Queensland, Royal Prince Alfred Hospital and Liverpool Hospital in New South Wales, Peter MacCallum Cancer Centre and MIA Monash in Victoria, Royal Adelaide Hospital in South Australia, and WA PET in Western Australia. And the Austin Hospital was able to achieve approval for a block grant to continue operations, and therefore 8 sites were to be involved with this Commonwealth PET data collection project. It obviously took subsequently some time for a number of the sites to actually purchase their cameras and commence operations. But over that period of time, it was necessary for us to actually generate a plan as to how this whole project would come together to enable the funding to proceed and patients to get access to PET scans for their clinical management. But around this time, there was also some quite interesting and very encouraging technology and radiopharmaceutical areas of progress. The initial PET scanners, as I mentioned, were quite clunky, had limited ability to perform regions of the body for acquisitions within timeframes that patients could tolerate on the scanner. But in 1998, the first PET-CT system was developed by Dave Townsend at the University of Pittsburgh and with CTI PET Systems. And in 2001, the first commercial PET-CT system was released by GE, the Discovery LS, followed very closely by Siemens with their Biograph and Philips with the Gemini camera. And these are shown here at the bottom of the slide. These were then subsequently installed at relevant PET sites around Australia, and certainly the images that were obtained from having PET-CT performed on the same imaging visit were fantastic, and certainly it was a lot easier than trying to chase down the films from the CT scan performed at another institution 2 weeks before they come for their PET scan, so you can actually try and interpret the scans. Also, in 2001, Cyclotech commenced operations for PET tracer production because clearly, as the number of PET sites went from 2 to 4 and then to 8, there had to be increased availability of FDG for clinical scans. And also plans for cyclotrons at other PET sites were developed. And that was particularly relevant for Western Australia because of the distances in shipping FDG. And the WA PET cyclotron facility was subsequently established. So this PET data collection project was aimed to be conducted over a 2-year period. It was agreed with the Commonwealth Government that demographic data collection on all funded patients over a 2-year period would be collected. And it was also then agreed that there would be prospective clinical protocols that would be multi-centre and look at management impact outcome would be conducted in 8 different cancer types: lymphoma, esophageal carcinoma, cervical cancer, soft tissue sarcoma, colorectal cancer, head and neck cancer, metastatic melanoma, and ovarian cancer, usually in the context of assessment of restaging of disease, except in the case of esophageal carcinoma and uterine cervix. The reason that these 8 cancers were selected is because these were the ones it was felt were firstly reasonably high in prevalence. Secondly, the evidence that had been generated so far in the literature and based on our own limited clinical experience suggested that there would be a positive result from management impact. And thirdly, that the recruitment would be sufficient for us to be able to get the numbers required during this period of time. I would mention that there was a concession by the Commonwealth Government that we did not need to perform studies in the staging of non-small cell lung cancer, solitary pulmonary nodules, or glioma, because they felt that the evidence that had been generated to date was sufficient for them to consider for that those indications for approval by themselves. And that actually happened over a subsequent 2 to 3 year period after we started developing this project. The Commonwealth MSAC process was agreed over the 2002 period of time, and the Australian New Zealand Association of Physicians in Nuclear Medicine, or ANZAPNM, and the Commonwealth Government entered into an agreement whereby the ANZAPNM would run this data collection project, and the agreements for that were finalised in 2003, during which time we had also been working on the data sets, the protocols, and the framework of how this project would continue. And I would mention this was to be the largest prospective PET data collection project ever undertaken when it commenced. I'd like to highlight a few of the key sites and individuals who are involved with this major project. The ANZ-APNM Management Committee was chaired by David McFarlane. I was asked to chair the protocol committee, which was responsible for the site credentialing, the development of documentation, protocols, also the conduct of the study, the collection and analysis of data, and the final reports. And then at the sites, they were led by Mike Fulham at Royal Prince Alfred Hospital, John Chu, Peter Lynn and Ivan Hoshon at Liverpool Hospital, myself at the Austin, Andrew Baldy and John Stuckey at MIA Monash, Rod Hicks at Peter MacCallum, Ben Kelly and David Wong at Southern X-ray Clinics at Wesley, Nat Lenzo and Peter Robbins at the WA PET Cyclotron Service, and Barry Chatterton, Dylan Bartholomew and Ian Kirkwood at the Royal Adelaide Hospital. A particular mention of Jane Ramshaw, who was the project manager, who was absolutely outstanding and essential for the design, the conduct and the analysis and the final reports for this major project. And also Janine Sargent, the CEO of the ANZ-APNM. This is an example of one of the PET demographic data collection forms that we assembled back in 2002, early 2003, where you would collect identifying information, obviously patients de-identified, but so we could follow the patients subsequently if required, disease categorization, indications to scan, what the results of conventional imaging were, either for staging or restaging, and how that was undertaken, their management plan, and what the post-PET stage was. So we could determine what impact on staging PET could have compared to conventional imaging and methods prior to the PET being performed. So data commenced for the demographic data collection in March 2003 and continued to April 2005, with the final report coming out in April 2006. And we analysed a total of 30,368 scans. These were consecutive patients at all sites over the 2-year period. Mean age of 50, roughly 50-50 male-female distribution. There were 29,585 oncology patients, 688 epilepsy patients, and only 95 cardiac patients. So you can see the very, very strong move towards oncology scanning by this time. Most sites had very similar numbers. Royal Prince Alfred Hospital, by far and away, had the highest number of patients during this time. The Royal Adelaide Hospital came on board late, which accounts for the lower number that they contributed here. Just looking at the oncology results, of those 29,585 consecutive patients, 4,000 were for diagnosis, 10,000 for staging, and 14,500 for restaging. They were distributed across most of the cancer types you would expect: lung cancer, SPN, colon cancer, lymphoma, melanoma, metastatic melanoma, the ones with the highest prevalence. And just looking at some initial results that we saw, looking at nodal staging in 8,000 patients, we found that we were able to upstage or downstage the nodal stage by well over a third of the patients, which clearly has impact on potential resectability and radiotherapy treatment planning. When looking at the metastatic staging, or M1 status, we found that in approximately a quarter of patients, they were upstaged or downstaged. But of course, the M1 staging doesn't completely reflect the number of metastatic lesions or where they're located, and that can impact on management. So at least 25% of patients from just a pure metastatic disease presence were changed in terms of stage by the FDG PET. But also, very interestingly, we had 15,000 patients who underwent restaging FDG PET. And almost 60% of patients had a change in their restaging classification, of which approximately 30% were either upstage or downstage specifically by the FDG PET. But impressively, almost a quarter of the patients had what was termed to be equivocal conventional imaging. So they just weren't quite sure if there was recurrent disease, and if so, exactly where it was. And the FDG PET scans in those patients were able to change from equivocal to either presence of disease or not. This is also reflected in this table, which you see at the bottom of the slide. So very, very impressive results in 15,000 patients. For the myocardial viability and epilepsy cohort, PET provided additional information that could impact on management in 1/3 of the patients in each of these cohorts. And in the oncology scans, as mentioned, a change in stage of disease occurred between 25 to 54.6% of all patients, which obviously has a potential for management change, but it was actually consistent across all tumor types and stages of disease because we had collected all of that data. And this was incredibly important data, which has subsequently been used as part of the MSAC reviews for oncology indications subsequently. Of the 8 prospective studies that was performed, I'll give one example, which was the impact of PET in colorectal cancer, which was look at how PET could change management in patients being assessed for possible recurrent disease, either because of residual structural lesion or they were about to go into possible surgery because they had apparently isolated metastases in the lung or the liver. And then clearly what those changes in management would have on patient outcomes. In summary, 196 patients were entered and were found to be eligible for analysis in the 2 groups I've mentioned, residual structural lesions or patients with pulmonary or hepatic mets which were potentially resectable. Importantly, management change was documented in 65.6% and 49.5% of patients within those 2 groups, with a high management impact in the vast majority of those patients, and additional sites of disease in just over 40 to 45% in both groups. And even looking at progression-free survival, so prognosis, on the basis of localized versus disseminated disease, overall or in patients with liver or lung lesions at the time of presentation, there was a clear, highly significant impact of PET in being able to stratify patients in terms of their prognosis. So while we were doing this, what was happening in the rest of the world? Well, clearly PET was expanding dramatically in the early 2000s, with government and insurance funding starting to emerge in the US, Europe, Australia, partially in Asia as well. A major event in January 2005 was when the Centers for Medicare and Medicaid Services, CMS in the US, expanded coverage for FDG PET to a broader range of cancer patients, which obviously made us in Australia very jealous because we were still in the middle of these data collection projects, whereas they were getting funding approval for a broad number of indications. But they did have to actually provide information to a data registry, which ended up being called NOPR and started in 2006. So they started a registry which was similar to our demographic data collection project 3 years after Australia started theirs. And they did not collect the full range of information that we did, but it was still an impactful confirmation for the subsequent more widespread general approval for PET by CMS, which was announced later that decade. And of course, PET-CT was being increasingly used and was almost the exclusive type of scanner which was being sold by that time. It wasn't just FDG PET that was being used in Australia. There were a range of other PET probes, fluorinated, carbon-11 related, and also O15, as I've mentioned. So really, the research in PET was starting to extend into other molecular imaging probes during the 2000s in a very productive and effective way. So how did this all come together, and what was the timeline for submission of these health technology assessments and Medicare approvals? Well, as I mentioned, the completion of patient recruitment occurred over a 2-year period, 2003 to 2005. From 2006 to 2007, submission of reports to the Commonwealth Government occurred. MSAC reviewed the PET indications between 2007 and 2010. And as a consequence, new Medicare items were approved for PET from 2008 to 2011. So that was the time frame. This led, once Medicare item numbers were approved, to an increase in numbers of PET sites from 8 to 12 in 2008. By 2009, there were 17, and by 2011, 27 sites around the country. And the number of PET scans went from 18,000 per year in 2006 to 45,000 Medicare funded in 2011 and 65,000 by 2017. So these submissions to the Commonwealth Government were pivotal for these Medicare item numbers being approved. And they also led to a range of high impact papers which were published in the major journals, one of which was our colorectal cancer prospective study, which I showed some data of previously, actually received the award as Clinical Paper of the Year in JNM in 2008. There were controversies and challenges, of course. Firstly, the initial Commonwealth Government review of PET in 2000 leading into 2001 caused a lot of controversy because the decision which was made wasn't entirely consistent with what the expert consensus was during the initial meetings. And subsequent challenges went on for some time, even while we were actually performing the planning and conduct of the prospective studies and demographic data collection. And although we have approval for the most common oncology PET indications, by the end of the 2000s, it took 10 years to actually achieve Medicare approval compared to when the original submissions occurred. And during that time, as I mentioned, CMS in the US started to expand even further. And by 2009, they had a very broad range of cancer indications that were approved. Canada and the UK also had fairly intense health technology assessments performed, which delayed their approvals quite considerably. And of course, in many countries, particularly low middle income countries, government support is actually very, very poor for PET even now. We still have many cancer indications that are not funded, and the HTA process is complex and takes considerable time. The other important point is that the costing for FDG PET studies by Medicare was quite controversial, so that the Medicare rebate established in 2002 was $953 for a whole body scan, which was based on a 1999 modelling of a non-capital based cost of PET. And no diagnostic imaging test has otherwise been approved where there hasn't been an inclusion of capital component. And in the last 20 years, we've not had that rebate amount changed at all, absolutely no indexation whatsoever. And a lot of independent modelling of PET costing, which has been done in Australia and submitted, has yielded very different results, but has not been successful in changing that rebate amount. There's also been many other HTAs required, such as breast cancer and rare and uncommon cancers, which has been approved finally for staging and will be eligible for rebate at the end of this year, with restaging assessment ongoing, which we hope will be not too much long after, which is essentially all other FDG PET avid cancers other than those that are currently approved. So incremental success, but again, 20 years after the original applications were put in. Compared to the rest of the world, Australia for PET access is doing quite well. In the Lancet Oncology Commission for Medical Imaging and Nuclear Medicine published last year that I was involved with, we performed a global survey of the number of PET scanners as well as workforce and other imaging instruments. In Australia, along with North America, some European countries and South Korea, has amongst the highest number of PET scanners per million population, 3.2 in fact, which compares to 1.23 10 years ago. So we do in fact have a reasonable number of PET scanners available for our patients around the country. And we are continuing with our health technology assessments. For metastatic breast cancer, for example, an initial application based on a meta-analysis, evidence of accuracy and costing model, which was created in 2012 in the Austin report, was submitted to MSAC in 2017 so that it could be used in patients with metastatic or suspected locally or regionally recurrent breast cancer was put in. And to give us some idea as to the level of detail which is now required for decision trees, this is the type of way you have to design your decision tree to show what would happen from an investigatory viewpoint, either with or without PET, what other investigations would be used, what the likelihood of a positive or a negative result would be, and what the interventions would need to be. This is then the basis for performing a cost consequence analysis, where you look at what the costs based on Medicare rebates of each of these particular treatments and interventions are, and from which you can actually work out what the potential savings would be, which in the case of FDG PET, we worked out in recurrent or metastatic breast cancer would be almost $8,000 per patient. This was part of the application, which was approved in 2019 by MSAC. And of course, during this time, we continue to develop new PET probes. The number of cyclotron facilities, hot cells and radiochemistry facilities in major and smaller hospitals is increasing. And our ability to perform multi-center trials through our collaborative and cooperative environment in Australia has been terrific. The trial groups through ARCNet, and interestingly, funding through philanthropic groups, government grants and industry has been ramping up, particularly as local biotech has been expanding over the last couple of years. And so really, this is an area of huge opportunity. And I know Ross Francis will be discussing this further in the next talk in this session. And finally, I would like to make mention of neuroscience, because this is an area in which Australia has been world leading, and it hasn't always had the same level of publicity, perhaps, as our oncology indications, which have a much greater impact and greater number of studies performed at all of our sites around the country. So a couple of key highlights that have occurred in the last 20 years are that, well, firstly, the first brain amyloid PET in Australia occurred in 2004 with carbon-11 PIB, and work which has been done by Chris Rowe with his colleague Victor Villamengue enabled the establishment of amyloid PET patterns in a range of different dementia types: amyloid, diffuse Lewy body disease, frontotemporal dementia compared to cognitively normal patients, in ways which matched and even exceeded those that were performed internationally. The first human studies with florbetaben, which is now FDA approved as a fluorinated amyloid tracer, were done in Australia. And also, very importantly, the rate of amyloid accumulation over time, and where its detection prior even to obvious clinical symptoms, and the time course from its appearing on an amyloid scan before true clinical symptoms that have impact on patient normal activities occurs, were established as part of the program of neuroscience investigation in dementia with PET here in Australia, and also tau imaging agents, which I think is really a highlight to the innovation in PET which has been performed in Australia over this time. So I thank you for your attention.